Hi, my name is Claudia Gonzalez, and this is my research project, the melanocytic effects of hipk2 CRISPR mutation, under the direction and supervision of Dr. Bethany Stanhope. Cavefish have developed the loss of pigmentation upon colonization of cave environments due to lack of sunlight, therefore making them excellent model organisms for research studies. hipk2 is a pigmentation gene known to have impacts in melanin production in zebrafish. Our hypothesis, by disrupting the function of hipk2 gene using CRISPR mutagenesis, we will find out what pigmentation impacts it has. Our objective is to disrupt the function of hipk2 gene using CRISPR mutagenesis and measure the impacts of pigmentation melanophore production. Image below on A, we find a surface fish, although this is our natural counterpart where it does receive sunlight and in a natural sea environment. But B, we have a cavefish which does not receive sunlight and has developed the loss of pigmentation and eyesight. Aim 1 is to characterize the impact of hipk2 pigmentation via CRISPR mutagenesis, anticipating that in the mutant fish we will have less pigmentation. Aim 2 is that by creating a high-resolution imaging analysis, we'll be able to characterize the loss of pigmentation, how many we've lost, our pigmentation depths, and melanophore count and size. Here below, we show our methods. We will do a 3% agarose PCR gel to make sure that our gRNA created has successfully mutagenized the zebrafish DNA. B and C will be shows the process of injection. D will be a 3-day post-injection analysis of the zebrafish embryo. And then E will be the full adult analysis with melanophore count, size, and pigmentation depth. For our results here, we show the anchoring of QTL, melanophore size, and cavefish genomes. A QTL is a quantitative locus when it shows a certain amount of impacts a pigmentation has on the zebrafish. We successfully localized that, and then we zoomed it to the zebra chromosome because we will be using zebrafish as our model organism due to its adaptability to lab environments as opposed to cavefish. We then sequenced the DNA of the zebrafish in chromosome 18 and created gRNAs on exon 2 to target the pigmentation gene. Here below, we did a multiple species alignments showing across zebrafish, cavefish, surface fish, humans, and mice, showing the impacts it has with the most impacts between cavefish and surface fish and zebrafish, as well the strongest mutations, making this a candidate gene. Below, we have a representative gel of the CRISPR genotyping, which will be done once the experiment is finished, with the A will be the control, the 2 will be the amplified control gene of the chromosome marker, and then 3 through 8 would be the successful mutagenization. Due to the experiment not being completed, we applied the anticipated results that we made with this gene called PMELA, which is a similar pigmentation gene, just on another chromosome. E and F we will use to measure not only the melanophore count, but its size as well as its depth in the future study that we will be finishing soon. Our conclusion is that we demonstrated a candidate way to phenotypically analyze pigmentation as shown above with PMELA. Our method will be applied to forthcoming experiments after we successfully mutagenize hipk2 in zebrafish. The identification of genes associated with color loss in cavefish will enhance our understanding in the pigmentation pathways with potential broader impacts into human pigmentation disorders. I'd like to give a special thank you for Dr. Evelyn Marquez Frazier and Nirthika Sutharakan. I'd also like to give another thank you to Office of Undergraduate Research Inquiry for giving me a grant to be able to fund my research. Jupiter Life Sciences Initiative, FAU Biological Sciences, and College of Medicine have been very helpful. Thank you so much.